Hello students, today we are going to study about non-Mendelian inheritance or such patterns of inheritance that do not follow Mendel's law of dominance, segregation or independent assortment. Let's start with incomplete dominance. As the name suggests, neither of the allele is able to dominate here and an intermediate type occurs. Let's take the example of snapdragon anterhenum. Here red flowers, capital R, was crossed with white flowers, a smaller. The genotype here is capital R, small r, and the phenotype here is pink instead of red or white. When these were swapped, capital R, small r into capital R, small r, we found capital R 1, capital R, small r 2, and a small r 1, 1 to 1 ratio. Here the genotypic ratio and the phenotypic ratio was the same. Law of dominance doesn't happen here. The second example is codominance. Here both the alleles assert themselves in a very dominant manner. The classical example is AB blood group, in which we have IA, IB, and both of them dominate. The other example is Rowan cattle. If we take their color, red color and white color, red color capital R, and white color W, capital W, we find in F1 generation capital R and capital W. That means both of them exist side by side. Both of them express themselves fully. Now let us consider the genotype and the blood group. If the genotype is IA, small I, then the blood group is A. If the genotype is IB, or IB, small I, the blood group is B. And if the genotype is IA, the blood group is IB. And if the genotype is I small I, then the blood group is O. Now let us move towards multiple alleles. Here what happens, two or more alleles exist in a gene. The classical example is ABO blood group, where we have three alleles, IA, IB and a small I. But the catch point here is that it occurs in a population, not in an individual. And only two alleles are expressed at the same time. Now the fourth one, pleiotropy. What happens in pleiotropy? One gene controls many traits. And the classical example here is sickle cell anemia, which is a classical example of point mutation, single mutation. What happens here? The glutamic acid here is replaced by valine at the 6th position of beta globulin chain of hemoglobin, due to which the RBCs become sickle shaped. Anemia occurs due to loss of blood, fatigue takes place, many organs get damaged such as spleen, brain, liver, lungs. Due to this spleen being dysfunctional, many infections occur. Another example is phenylketonuria. What happens here? Mental retardation takes place and skin pigmentation occurs. The third one is Marfan syndrome, due to which long limbs occur and heart disease also takes place. What happens in polygenic inheritance? As the name suggests, many genes control traits and each is additive in nature. That means add something. For example, human height, human body weight and human skin color. As a matter of fact, human skin color is controlled by 3 pairs of genes. That's polygenic inheritance for you. Now let us move to epistasis. What is epistasis? Here one gene masks or suppresses the other. If it masks the other one located on another locus, it's known as dominant epistasis. The classical example is fruit color in squash. Here the F2 ratio is 12:3:1, 12:3:1, and when one gene it suppresses, doesn't mask, it suppresses another gene, then it is known as recessive epistasis, as we see in the coat color in mice. Here the F2 ratio is 9:3:4, 9:3:4. Now let us move towards linkage. The classical example here is Drosophila melanogaster, and this experiment was done on the fruit fly Drosophila melanogaster by T. Morgan. He observed that there are two traits here, or the characters. First, the body color, gray or black. Then the wings, normal or vestigial. Both were closely related to each other, and they were on the same chromosome. As a result of that, both of them couldn't assort itself, and they got inherited by most of the offsprings. So if the linkage is strong, a recombination doesn't occur. But if the linkage is incomplete, recombinations occur. Now let us take the example. Suppose that there are A and B genes located on the same chromosome. What happens here? If they are very closely related, strong linkage, no crossing over is there, and as a result of that, there is no recombination. But if they are very far, crossing over takes place, weak linkage, and as a result of that, what happens? Recombinations frequently occur. Now let us move towards the last topic, sex-linked inheritance. When genes are located on sex chromosomes, they show a unique pattern, as observed in the case of hemophilia, color blindness, or Duchenne muscular dystrophy. These are sex-linked recessive. Here, the most affected ones are the males, because they have only one X chromosome. The daughter here act as carriers, and there is no transmission from father to son. There is crisscross transmission. What happens? The X chromosome is transferred directly to the daughters instead of the son. The daughters act as carriers, and they transfer it to the grandson. So we have studied all the relevant topics related to the exceptions of Mendelism or Mendel's law of inheritance. So it's Happy New Year to all of you. Enjoy yourselves, have a blast, and like my videos too. And if you haven't subscribed my channel, do it now. So on this note, I would like to close this particular video or lecture. Thank you. Good night.